Hey everybody, we are moving on. We're going to take a look at plant responses to both internal and external signals or stimuli. All right, so a question. Do plants sense and respond to their environment? And the answer is yes. They are able to adjust their growth and development patterns in order to respond to their environment. So what you're looking at here is a plant grown in dark versus a plant grown in light. And you can see that this is the plant that was grown in light, and it's much more organized than the plant that was grown in the dark. So a quick comment. Plants can't move away from a stimuli the way that animals can, but they can change their growth response. And so what that means is plant bodies end up being much more flexible in morphology than animals. We're going to talk a little bit about cell signaling in this chapter, and so if you're really struggling to remember that, I do suggest you guys review chapter 11. Just to refresh a little bit. So a classic example is with phototropism, and this is the plant growth response to unilateral light. And so the observation that was made is that plants will bend or grow towards a light source. So we've got several different experiments with phototropism. And the mechanism for phototropism is that cells on the dark side of the plant are going to elongate faster than cells on the light side. And so what that means is that it can bend towards the light. So you've got this uneven growth rate that will cause a bend in the stem. So now a quick question then would be, what is the adaptive value of that, of growing towards the light? And it turns out that it tilts the leaves toward the light source, and so it's more efficient for photosynthesis. So now we need to know what causes phototropism. So we've got a chemical messenger from the tip of the plant that causes the growth response in the stem, and then the distribution of the chemical changes in the unequal light, and so it results in unequal cell elongation. So here we are looking at a plant cell. If we kind of zoom in on the cell wall here, you can see that we've got some auxin, which is a plant hormone that is stimulating the proton pump. And so when that happens, you've got expansions within the cell membrane. So the cellulose is loosening, allowing the cell to elongate. Here are some of the classic examples of the experiment. So we've got the Darwins in 1880, and they had a control, so they didn't do anything to this plant. You can see that it bent towards the light. They had a plant where they removed the tip of the plant and the plant grew straight. They had a plant where they covered the tip of the plant with an opaque cap so no light could get through, and the plant grew straight. They did it again with a clear cap. The plant bent. Then they covered the base of the plant, and the plant still bent. Then we've got the Boysen-Jensen experiment in 1913. And what they did here was they removed the tip of the plant and they sort of separated it with a gelatin block and then put the tip back on. And so that gelatin block separated the tip from the rest of the plant, but it still allowed the hormones to travel through. And so the plant bent. And then they separated it by mica, and this did not allow the hormones to travel from the tip through the rest of the plant, and so the plant grew straight. Then we have the Went experiments, and with this one they took the plant and they excised or cut off the tip, put it on an agar block. The auxin is going to diffuse into the agar block, and so what they did then, they had the control with the tip removed, they had a control with an agar block that had no auxin, so there was no effect. Then they put it back on, and this time they allowed the auxin to travel to the whole plant, and so it continued to go straight. Then they offset the agar block, and what that did was it only allowed the auxin to go to one side of the plant, and so it caused a curvature, a bend in the plant. And so what I've been talking about is that auxin is a plant hormone. So a hormone is a chemical signal that's produced in one location, and then it's transported and has an effect in another location. And so phototropism is caused by a plant hormone. Now we've got a few plant hormones we're going to look at pretty quickly. They're produced in small quantities, and their effects reflect a balance between several hormones. So we have 5 classic plant hormones that we're going to talk about. We have the auxins that were named by Went in 1926. This was the first plant hormone that was described. Again, it helps cell elongation, but also fruit development, apical dominance, tropism responses. It stimulates root formations, and it can be used as a broadleaf plant killer. Let's see. And so where is it produced? It's produced in the apical meristems, in young leaves, and also in embryos. So apical meristem means like the top of the plant where the growth is happening. Then we've got the cytokinins. These were first isolated in coconut milk or endosperm in the late 1940s or in the 1940s. And cytokinins were named because they stimulate cell division. So again, their major effects: stimulating cell division, delays senescence, helps with root growth and differentiation, and it's produced in the roots. So when you look at the auxin versus cytokinin ratios, that's what's going to control shoot or root differentiation in tissue cultures. Then you've got gibberellins. These were found in the foolish seedling disease in rice. We have like 70 different types of these that are known. This helps with internode elongation, seed and bud germination, flowering in some species, and also fruit development. Internode elongation is going to be this space, so like you've got the nodes where you've got leaves, and so the internode elongation would be like the space on the stem between where the leaves come out. And so this is kind of what happens when you have gibberellins versus no gibberellins. Again, produced in apical meristems, young leaves, and embryos. Then we have abscisic acid. This is going to slow or inhibit plant growth. This is a stress hormone that's going to be produced by the plants in unfavorable conditions. And so if conditions aren't favorable, it's going to slow down the plant growth until conditions improve. So again, inhibits growth, allows for seed and bud dormancy. It closes stomata, and also leaf drop as well. And so this is going to be produced in leaves, in stems, and in green fruit. Next, we've got ethylene, and this is a gaseous hormone that has really fast diffusion rates. It's often going to interact with the auxin, and it helps with fruit ripening. It accelerates senescence, and then also stem and root elongation. And this is produced in ripening fruits, senescent tissue, and also nodes. So a new hormone is the brasinosteroids. This is a steroid hormone. It's actually similar to animal sex hormones, and this functions in cell elongation and division in stems and seedlings. We also have commercial applications of plant hormones. They can be used as weed killers, helping with making seedless fruits, rooting of cuttings, and also tissue culture. All right, so now we want to talk about plants and how they respond to light. So responses to light are really critical for plant success. Plants can detect the presence of light, but also its direction, its intensity, and its wavelength, and that's all really important. So we've got two major light receptors. First is you have the response to blue light, as in phototropism, and so that was again the bending. But you also have a response to red light, as in photoperiodism, and this is essentially the physiological response to changing in day length. So phototropism was the bending in response to light. Photoperiodism is how plants respond to the length of the day, or the amount of the length of time that they get light exposure. And so the advantage is that it matches the growth of the plant response to the proper season. So like it makes sure that leaves will drop in the fall, or that the flowers are produced in the right time of the year for mating, things like that. Now we've got different types of plants that respond to lights differently. So we've got short day plants, long day plants, and then day neutral plants. So short day plants flower when days are shorter than a critical period. And really what we're looking at here is those are going to be long night plants. So mums and poinsettias are a good example. Then you've got long day plants, and these are going to be plants that flower when days are longer than a critical period or have shorter nights. Examples here would be spinach, iris, and lettuce. And then there's day neutral plants, and these are going to flower whenever they have enough energy, and it's not dependent on the length of the day. So examples would be roses or African violets. So night length is actually what appears to be really important here. So it's night length that actually controls the flowering response, not the day length. And the proof is that experiments show that if you sort of interrupt that dark period or night period with a couple minutes of light, it actually resets the clock. And so that is what happened here. So here we have the hours of light and hours of dark. And as soon as you kind of look at this, you're interrupting the night with some red light, and what happens is it keeps the plant from flowering like it should. So it's the relative length of night that is critical. So it's the length of night. It doesn't have to be absolute, but it's relative for the response to be triggered. So what detects day and night length changes? And it's actually phytochrome. And so it's a plant pigment that's involved in photoperiodism, and it is a red light receptor. So you've got two forms of phytochrome. You've got the PR, or the phytochrome red, which responds to red light, and then the phytochrome far red, which responds to far red light. And so it can change between the two forms, and it's the ratio that you're looking at, or the accumulation of enough of the phytochrome far red that triggers the responses. So in red light, the phytochrome red will convert to the phytochrome far red, and it allows responses for things like seed germination and flowering. So photoperiodism is very sensitive. It could take as little as a 1 minute difference, and it essentially sets the clocks for plant responses. It also affects seed germination, which you can read more about in the textbook, but also stomatal opening and leaf drop. All right, so now the last thing we're going to talk about are plant responses to herbivores. So plants have many physical and chemical defenses against herbivores. So keep in mind that if something's trying to eat a plant, the plant can't get up and move away from whatever's trying to eat it. So it has to respond in other ways. So an example of a physical defense would be something like thorns. A chemical defense would be crystals or tannins or other toxic compounds, or even just compounds that make the plant taste really bad. So herbivores will often trigger a plant to release chemicals to attract predators or to warn other plants to increase their production of toxins. So what we have here, we're looking at a leaf. This leaf is being wounded. See, we've got a caterpillar eating it. And so the first thing that happens is you've got the plant being wounded. And you have a chemical in the saliva of the caterpillar, and both of those things are going to signal a transduction pathway. That pathway is going to cause the synthesis and release of volatile attractants. Once they are released, essentially then what's happening is you're recruiting these parasitoid wasps that will then lay their eggs within the caterpillars. All right, and so essentially this caterpillar is wounding the leaf. That's starting a signal transduction pathway, which attracts a wasp, which will then get rid of the caterpillars. So we also have pathogens that can affect plants. And so the first defense of a plant is going to be the epidermis. You're going to have this layer that is going to try to block out any pathogen. Then the second defense is going to be a chemical event to either restrict the pathogen or kill the pathogen. And so what we are looking at here is you have a pathogen. So we're looking at two leaves on the same plant. So you have a virulent pathogen. Okay, that pathogen has, you can see this leaf does not look healthy. It's affected this leaf. It starts a signal transduction pathway. And so we have this hypersensitive response. So the plant is then going to send, this leaf is going to send signals out to the rest of the plants. And the rest of the plant now will have this acquired resistance because this leaf was attacked by this pathogen. Okay, so that's it for this chapter. Things you need to know. The general plant hormones and their effects, so the things we talked about. You guys need to know phototropism, photoperiodism, and then you need to have a general idea about how plants respond to stress of herbivores and pathogens. All right, and so your lecture question for today is, please explain in your own words how plants are able to respond to things like light, hormones, and predators if they're unable to remove in response to such things. All right, so please send me a message via the class website with your response to the lecture question. Thanks, guys.